much. Go ahead, Joanne, please, with the intro.
Okay. Well, I'm Joanne Jessen, and I'm really pleased to be able to introduce Dr. McCray to all of you today. I happen to be a patient at Hopkins in their CMT clinic, so this is certainly a topic near and dear to my heart. Dr. McCray is an assistant professor of neurology at Hopkins. He's co-director of the CMT disease clinic. He graduated from the University of Pennsylvania School of Medicine, and his major interest is the overlapping disease mechanisms in hereditary and acquired neuropathy. And he is going to talk to us and hopefully stimulate lots of questions from us, but also increase our understanding of why genetic testing is important and why, even though we may have a diagnosis of idiopathic, why it might be important to follow through with some testing. So without further comments, I'm just going to turn this over to Dr. McCray. And I'm happy that Pat and Steve and Judd will be helping me monitor, because this is the first time I've done this. And so I'm keep wondering if I'm going to remember to check in. So anyway, Dr. McCray, go for it.
Thanks, Joanne. So I just hit share, and hopefully you can see my slides filling the screen now.
Yes.
Good. Okay. So thanks for the introduction, and thanks for the invitation to speak today. So as Joanne mentioned, I'm going to try to give you sort of a perspective on some of the ideas that have come from research over the years that sort of link up the idea of hereditary or inherited neuropathy with acquired forms of neuropathy. But when I say acquired, I mean things that are not genetically determined. So that would include idiopathic neuropathy, diabetic neuropathy, chemotherapy-induced neuropathy, and so things that you sort of develop in life independent of genetics. Although I'm going to sort of give you the perspective that maybe there's something to be said for some genetic risk factors in some of these other forms of acquired neuropathy. So as was mentioned, I did give a talk recently to the FPN, and so some of you may have tuned in for that. So I apologize. Some of the intro is similar. It's actually identical to that talk. So if you saw that talk, then you can take a little snooze for this first part. But I promise you that there are different ideas and different slides in here, but I wanted to make sure everyone's kind of on the same page from the beginning. So just to kind of give a perspective of how I think about neuropathy, we sort of think of the nervous system as the central nervous system, which is the brain and spinal cord. And then everything outside of the brain and spinal cord is the peripheral nervous system, and peripheral neuropathy affects that part of the nervous system. So it affects the nerves after they've left the spinal cord. And I think hopefully you can see my arrow when I point to things. You see that sort of circling? Okay. So as I'm sure you know, there's sort of different manifestations of neuropathy. You can either have neuropathy that affects the sensory nerves, the motor nerves, or both. And when it affects the sensory nerves, you can get a loss of sensation manifesting as numbness, or you can get any number of sort of extra, unusual, uncomfortable sensations like tingling, pain, electrical shocks, heat, etc. And then when neuropathy affects the motor nerves, that manifests as weakness. And when it gets more severe, you start to see atrophy of the muscles that are the target of particular nerves. And so symptoms in peripheral neuropathy, whether it's caused by a genetic process or whether it's something like diabetes or anything else that causes neuropathy, usually follows a pretty typical pattern. And that pattern is that it's usually very symmetrical, meaning that it affects both sides of the body equally. And then it's what we call length dependent. And so what that means is that it affects the longest nerves first, and those are the nerves that go to the toes and feet. And then over time, the sort of length dependent rule is that shorter nerves start to get affected. So initially that might manifest with numbness or tingling or pain in the toes. And then over time, if it gets worse, it would sort of move up into the shins and then maybe up to the knees. And around that time, roughly, is when hands might be involved, because the nerves to the hands are about as long as the nerves that go to the knees. So that's sort of typical progression. And so just to step back a bit, what's actually happening here? What are we talking about when we talk about peripheral nerves? And so nerves are basically, in a very simplistic analogy, sort of a conduit for electrical signals. So either they're sending electrical signals from the brain spinal cord out to the muscles to cause the muscles to contract, or they're relaying signals from the skin to send sensory information back up to the brain. And so in this analogy, the wire of this electrical pathway is really the axon of the nerve shown here in this long segment. And so this particular picture shows a small cell body and an axon going out. And so in reality, for peripheral nerves, this cell body is really tiny with respect to the axon. So the axon can be like a meter long, and the cell body is just a tiny little speck. So most of nerves are really made up of axons. And then around each axon is insulation, which is known as myelin. And the myelin has these gaps in between it. So peripheral neuropathy can affect either the myelin or the axon or both. And when it primarily affects the myelin, we call it a demyelinating neuropathy. It's shown here. You can see sort of the axon is intact, but the segments of myelin in certain places have been stripped away. And that causes a slowing of nerve impulses, but generally not a reduction of the nerve impulses. And then axonal neuropathy happens when the axon itself is damaged, but the myelin sheath remains intact. And so this generally happens at the end of the axon. So it kind of out where the axon makes contact either with the skin or with muscle, and then slowly works its way back. And so again, this is another reason why neuropathies are what we call length dependent. So they affect the end of the axon first and the longest axons first. So when I see someone with neuropathy, one of the big questions is, what's the cause? Is this something that is due to a vitamin deficiency, diabetes, or is this something that's actually genetically determined? And so in favor of something being inherited or genetic is if there's a family history. That means if other people in the family are affected. And the people that are most informative would be siblings, children, or parents. And so if you start hearing that multiple family members are affected with neuropathy, that sort of raises suspicion that this is an inherited process. The age of onset is another big contributor. So what I'm going to talk about is mostly Charcot-Marie tooth disease, but in that disease, while there's many different kinds, most people have some sort of symptoms in their teens or 20s. There are late onset forms, which we define as after age 40, but for the most part, it's an early onset disease. Often people notice changes in the way their feet look, which is sort of a clue. And then even if people don't really notice overt symptoms earlier in life, if you dig enough, you might find that maybe they were not very athletic or kind of clumsy when they were younger. And so this indicates kind of a really long-standing process that was present from birth because it was actually inherited. And then when we do an exam, we often see a lot of atrophy of muscles, and then sort of typical neuropathy signs like sensory loss, difficulties with balance. And then we really look for these changes in the way the feet look. Sometimes, but most of the time not, the EMG or nerve conduction test can really indicate an inherited neuropathy. And that's mostly true with demyelinating neuropathies. They have a very specific pattern when they're genetically caused. But for axonal neuropathies, there's not really a good signature that we can see on nerve conduction studies that tells us whether it's likely to be an inherited or non-inherited process. Things that certainly argue against an inherited neuropathy are if the symptoms come on really rapidly. That generally doesn't happen. And then if there's a known other reason to develop neuropathy, then you don't really have to search for an explanation if there's already a clear explanation. A lot of asymmetry is also another sort of sign that there might be something else going on. In particular, back and spine problems can manifest with the same kind of numbness and weakness that you get with neuropathy, but usually that's asymmetrical. And if you do an MRI and see a lot of sort of degeneration and arthritis in the back, that can be an alternative explanation. So briefly, just to talk about the way inherited neuropathy can move through families. So it can be inherited in a number of different ways. So there's dominant disease, which is by far the most common way that neuropathy is passed through families. And this just means that you only need one abnormal copy of a disease gene to get the disease. So if you yourself have a neuropathy that's genetically encoded, then you have about a 50% chance of passing that on to children. And in that case, it's usually pretty obvious when these are running through families because there's multiple affected family members, because about roughly half of them have the disease. And then there's other forms where you need two copies of the disease gene, and these are recessive forms. And this is the exception in inherited neuropathy. And it's also hard to pick up because you need 2 copies, one from each parent. The likelihood of that happening at multiple people in the same family is lower. So these are a little harder to notice in a family. Then there's X-linked forms, which are a little more complicated. They sort of get passed down in a complicated way, depending on whether the person that has the disease gene is a man or a woman, and they sort of can almost look like they skip generations. But then the other twist is that you don't necessarily have to have a family history to have a genetic neuropathy. So some genetic mutations happen spontaneously, so you can be the first person to have that mutation. So the only person in the family that has it, you would then be at risk for passing it on to children, but the fact that parents or siblings don't have the same neuropathy doesn't necessarily rule out an inherited process. So just a bit about how we classify neuropathy, and it's to be honest, it's a mess, and this is partly historical because a lot of the names were given before genetics was really well developed. So for the most part, if a genetic change causes only neuropathy and nothing else, we call it Charcot-Marie-Tooth disease, and that's named after these 3 gentlemen here. And there's type 1, which is demyelinating, and type 2, which is axonal. There's also, so we call it CMT when there's sensory and motor neuropathy. Then there's also inherited forms that only cause sensory neuropathy or only motor neuropathy, and those get different names. I personally try to call them all CMT because I think the genetics has really showed us that the same gene can cause CMT1 or can cause, say, CMT2 and HMN or HSAN. So looking at the genetics, it seems that it's kind of messy, but the same gene can give different sort of manifestations of neuropathy. Then there's other inherited conditions where neuropathy is just one part of other symptoms. So there might be heart problems or other brain or spinal cord problems. And in those cases, we don't call it CMT. They get different names, like, for example, familial amyloid polyneuropathy or giant axonal neuropathy. So just to give you sort of a sense of the messiness of hereditary neuropathies in general. So I think one big question maybe from this group and from people that have non-inherited neuropathy is, why does someone like me study neuropathy, CMT and inherited neuropathy? What do I hope to gain from that, or what is to be gained? And so as far as sort of uncommon diseases go, CMT is not so uncommon. It's actually affects about 1 in 2,500 people. And so that's still pretty low, but that makes it the most common inherited neurological condition worldwide. So relatively common amongst uncommon things. But the other thing is really that the hope is that CMT can teach us a lot about general nerve health and disease and have implications far beyond patients that have CMT. So one thing that's really tough about, say, diabetic neuropathy is really getting a foothold on how to study it, or even idiopathic neuropathy is even harder. How do you understand what's going wrong when you just don't know where to start? And so, and the other assumption here is that, and I think this is borne out by research, is that the underlying process or what's going wrong in the nerves in CMT might be very similar to what's going wrong in diabetic neuropathy or idiopathic neuropathy. And so CMT can kind of be thought of as nature's experiment. So what's happening if we understand which genes are causing CMT, it's basically, a sign that these specific genes are really important for nerve health. And so it gives us a foothold on where to start the research. So of this list on the right, all these genes cause CMT. And so we know that they're all important for some aspect of what keeps nerves alive and what makes them healthy. And so if we study those, then maybe we can get a better understanding of nerve health, and that might be applicable to other forms of neuropathy. And then in the bigger picture, there's lots of ways to injure nerves beyond just peripheral nerves: strokes, traumatic injury to the brain and spine, exposure to toxic substances. These are all ways to injure different nerves in the body. And the commonality is we don't really have any treatments for any of them to either protect from injury or to help the nerves recover. And so again, CMT research is a good way to kind of really get a handle on some of these disease processes that we might be able to eventually treat in some way to help a lot of people, not just those that have CMT or peripheral neuropathy. So at this point, there's over 100 different genes that are known to cause CMT. And if you do genetic testing, there are different panels that will test a number of these. And so the one that we use most at Johns Hopkins, which is currently free through Invitae testing, tests for 102 different genes. So it's a pretty good survey of at least the known genes that are out there. And with CMT, with these over 100 forms, there's really 4 types that make up a big chunk of that. But then there's, a very small piece of the pie for these other, 100 or so genes. But what we've learned from the past 20 or 30 years with gene discovery and then studying what these genes actually do is that the genes in CMT are involved in many different aspects of neuronal biology and function. And so these are some of the pathways that are affected by CMT. And so here again, you have the cell body of the nerve, the axon and the myelin, and then the end of the nerve. And so as far as CMT type 1, which is the demyelinating form, what really has been shown by the genetics is that all of these genes affect something to do with myelin. So myelin's made by Schwann cells, and all of these genes do something to disrupt the myelin formation or its health in some way. In CMT2, there's a lot more variability in the sort of problems that happen. And so 2 main categories are sort of genes that are involved in the structure of the axon and then movement of things through the axon. And so as far as the structure, so this we call the cytoskeleton. And what you can think of is, so these are things like these neurofilaments here and microtubules. And what they do is sort of create the scaffold of the axon and give it shape and structure and sort of give it strength. And so we know that genes that are directly involved in providing that structure to the axon can cause CMT. And then the other aspect, other sort of pathway that's been highlighted by these disease genes is intracellular transport. And so what happens along particular microtubules is these little motor proteins that kind of land on these, they're sort of like tracks, and they move things back and forth throughout the axon. And so there's a lot of genes that are actually motors that are supposed to be driving things essentially from one end of the axon to the other. And so this really highlights the importance of this process for neurons. And you can imagine why, because in reality, this is 1 meter long for some nerves. And so to get something from this part all the way out to the end of that is a huge challenge. And that's a really unique challenge for nerves as compared to other cells of the body. And so it's no wonder that a subtle disruption of that problem might create a nerve disease. And then sort of as a corollary of that, there's a number of genes involved in what we call vesicular trafficking. And so these are basically genes that are involved in sort of sticking to these motor proteins and helping very specific things move back and forth. So a lot, these genes really point to the structure of the axon and movement of things throughout the axon. And then there's also a lot of genes that are involved in mitochondrial biology. So these things here are mitochondria, and they're the energy producers of the body. And again, these are particularly important in neurons because the whole process of transmitting electrical signals back and forth constantly uses a lot of energy. And so neurons are really dependent on very careful regulation of mitochondrial function. And so these genes affect everything from the movement of the mitochondria to how they come together and how they split apart. So there's many different genes that affect mitochondria particularly. So, so one of the sort of connections I think that's interesting between inherited neuropathy and acquired neuropathy comes through the study of chemotherapy-induced neuropathy, or I think I abbreviate as CIPN throughout the talk. And so we know that there's many different chemotherapy agents that can cause neuropathy, but a lot of them directly have an impact on these microtubules, which are these sort of tracks that things drive along within the axons. And so this is kind of a blown up version of a microtubule with these motor proteins kind of walking along and carrying things to and from the sort of cell body or the end of the axon. And so 2 drugs in particular that are very well described to cause CIPN are paclitaxel and vincristine. And it's known that these both directly bind to microtubules and change their function in a way that we think is really central to what causes the neuropathy. And so this is just a picture of a neuron in a dish that actually does kind of look like this one on the left. This is a sort of normal one. And this in green is a marker of some of these microtubules. On the right, the same sort of cultured neurons have been exposed to paclitaxel. And you see this axon now looks unhealthy. It's sort of irregular, has these sort of blebs in it. And the microtubule marker here looks very different. So we know that these drugs directly affect the microtubules and the axons and make neurons sick. So one of the big questions in CIPN and often other forms of neuropathy is, why do some people get this neuropathy and some don't? So we know people can be exposed to the same dose of chemotherapy, be roughly the same age and be of the same general health, and some people get it and some people don't. And so one sort of interesting finding that I think speaks to sort of genetic susceptibility is that patients that have CMT type 1A seem to be really susceptible to CIPN. And when they get it, it's really severe. And so what that suggests is that there's something about that genetic background, that genetic change that they have that is really puts you at high risk of what the chemotherapy drugs are doing. So it does kind of suggest there's an overlap between the way nerves are sick in this type of CMT and the way nerves get sick with CIPN. There's also been a lot of studies to try to look at people who get bad CIPN versus that don't, and try to understand, how are they different? Is there something genetically or otherwise that puts people at risk? And so some of the interesting findings have been that some of these same pathways in CMT show up in people that seem to be at high risk. So a couple of genes where they found subtle changes are these two I've listed here. And these are both genes that are involved in sort of the cytoskeleton that I mentioned earlier. It's sort of how the axon is structured. And there's also been a specific study of people that develop CIPN to look at specifically whether they have any changes in CMT genes. And so there's a partial panel of those genes here. So what they were looking for is not mutations that cause CMT, but maybe subtle changes that in these same genes that just impart a little bit of increased risk. And what they found was that there were two genes, this PRX and ARHGEF10, both of which cause CMT, but these showed up as there were certain changes in these genes that some people have that put them at increased risk for CIPN. And so what this suggests to me is that some of the same disruptions that people get with genetic forms of neuropathy might be happening to some extent in other people. Those genetic changes aren't enough to cause neuropathy on their own, but then when you put on exposed to some toxin or some other stress, then the nerves become sick. And so it really suggests there's an underlying similarity in the risk for certain kinds of neuropathy and what people have with inherited forms of neuropathy. So another example is this gene that I happen to study in the lab. It's called TRPV4. And so this is a gene that is mutated in CMT type 2C and also causes sort of related diseases called spinal muscular atrophy. And it's an ion channel. So this is what it looks like. It basically sits on cells and it can open and allow things to flow in, and it can close. And the mutations that cause disease tend to open the channel more and allow more, in this case, calcium to flow into the cell, and that is somehow toxic to nerves. And so this has been studied in CIPN actually, and it's been found that if you block TRPV4, we've shown that if you block TRPV4, it can help these patients most likely, at least in animal models. But a different study showed that if you block TRPV4, it might actually reduce the risk of CIPN, at least in a mouse model. And so this is the data from that paper where they basically looked at the size of the response from a nerve in the tail of mice. If they gave paclitaxel, that response went down, but if they at the same time inhibited TRPV4, that response, that drop in response was lessened. So this was actually sort of a protective effect. And they saw it even more 2 weeks after treatment. So this suggests that TRPV4 that causes CMT might actually be doing something bad in CIPN as well. And there's been a number of papers that sort of expanded this kind of observation to include other nerve injury conditions. So TRPV4 inhibitors, and at least this is all in mouse models or rat models at this point, but has been shown to be helpful in stroke, whether it's an ischemic stroke where there's a lack of blood flow or a hemorrhagic stroke where there's bleeding in the brain. And then there's also some evidence that TRPV4 blockade is helpful for spinal cord injury. And then some of the work we've done with a collaborator shows also in traumatic brain injury that if you add a TRPV4 inhibitor, it can reduce the sort of size of the lesion that happens after the injury. So it's kind of an example of studying a disease gene in CMT and then finding that it might be really useful in other conditions well beyond CMT and even peripheral neuropathy. There's another story that's come from CMT literature that I want to highlight. And so this is another kind of microtubule focused idea. And so I mentioned microtubules before, but so these are really kind of complex dynamic things in the axon, and they can be modified in certain ways. And one of the modifications is what's called acetylation, where this sort of kind of gold glob here gets added to the inside of this barrel of microtubules. And when this happens, it makes the microtubules more stable. It helps things move along them. And axons in particular have a lot of acetylation in them. And it's been shown that in multiple different types of CMT, the level of acetylation of these microtubules gets decreased. And it's known that there's sort of this dynamics between acetylated and deacetylated tubulin, and that HDAC6 is a gene that sort of promotes deacetylation. And so what they've seen is that if you inhibit this gene or this protein HDAC6, it sort of restores the normal level of acetylation in these animal models of CMT, and that actually can improve their neuropathy. And so this is sort of the CMT2F model, CMT2A model, and they've even now gone on to test the same idea of sort of fixing tubulin acetylation in axons in chemotherapy-induced neuropathy, and they've again seen a benefit in animal models of that condition. So it's sort of another example of an insight that came from studying CMT that might be more generally applicable to other forms of neuropathy, probably particularly those where there's a problem with moving things, this axonal transport within neurons. Sorry, how am I doing on time? I'm not, hopefully not talking too long. So this was the last example I wanted to go through of this sort of connection between genetic neuropathy and acquired neuropathy. So this was a recent publication just in the past year, identification of mutations in this gene called SORD that cause recessive CMT. And it turns out that I mentioned that recessive CMT is less common than dominant, but it turns out that this SORD mutation is probably the most common reason to have recessive CMT. And so there's probably a lot of people out there that had CMT that didn't know the gene, and now this might be the answer. And so it turns out that this SORD gene is actually an enzyme that's called sorbitol dehydrogenase, and it functions in this polyol pathway. And so this pathway is a metabolic pathway by which glucose gets converted to sorbitol, and then the job of SORD is to convert sorbitol to fructose. And so if you don't have SORD, you build up sorbitol. And so that's exactly what they see in patients that have these SORD mutations. They have more sorbitol circulating in their blood and probably also in their cells, in particular neurons. And what they did was remove SORD from fruit flies. And when they did that, they developed this sort of degeneration of their eyes and their retina. So this is basically the mutant here, where you can see these kind of holes forming in the Drosophila eyes. And then they could actually block this enzyme here to try to reduce the accumulation of sorbitol, and that made the flies better. So it really suggests this might be a good strategy for these patients, which is really exciting. But the other really interesting aspect of this is that for a long time, the polyol pathway, this sorbitol dehydrogenase pathway, has been known to be important in diabetes. So we know that in diabetes, there's elevated glucose. When glucose goes up, this pathway sort of gets activated, you get more sorbitol and you get more fructose. And patients with diabetic neuropathy actually have elevated sorbitol, similar to these patients that have SORD neuropathy. And so there's been a lot of interest in trying to inhibit basically the generation of sorbitol by inhibiting this aldose reductase step of this pathway, which is the same thing that they did in these fruit flies. And so there's been a lot of studies. There's certainly not sort of game changer studies, but there is some evidence that targeting this particular pathway can actually improve things somewhat in diabetic neuropathy. And another interesting aspect of this is that, it sort of like I was pointing out with CIPN, why some people develop diabetic neuropathy and others don't is a big mystery. Some people can have really high glucose levels and not get neuropathy, and some can have just a small elevation of glucose and get a really bad neuropathy. And so, why is that? And so there's been studies also to look for genetic risk factors for diabetic neuropathy. And one of the genes that has come up is actually this aldose reductase gene that's part of this polyol pathway. And so it really suggests there's kind of a convergence of the idea that this pathway is important, not only in inherited neuropathy, but maybe also in diabetic neuropathy. So, so hopefully that gives you some sense of how I see connection between inherited and other forms of neuropathy. So one idea is that the underlying problems with respect to what's actually going on in the nerves might have a lot of overlap. And there might sort of be an accumulated genetic risk in idiopathic neuropathy. So maybe you don't have one gene that gives you neuropathy, but you have several changes in many different genes that generally impart an increased risk and make your nerves a little more susceptible to the normal wear and tear of life, or to a little bit of elevation of glucose, or exposure to a chemotherapy drug. And the other idea is that hopefully, since it's hard to study some of these other kinds of neuropathy, it's actually easier to study CMT in a lab, because we know exactly what we're dealing with. We can give a mouse essentially CMT and study it. And so the idea is that if we study these genes, study these patients, that might lead to treatments not only for them, but for other kinds of neuropathy. And so just to give you a sense of, some of where some of these studies have led. So this is the TRPV4 inhibitor that's been used actually in unrelated clinical trials for heart failure and chronic cough, but we think it might be something that down the road could prove to be useful for other conditions, neuropathy, and maybe other kinds of nerve injuries. And then this is an HDAC6 inhibitor that's been used in clinical trials, again, for other conditions, but might someday turn out to be useful for different kinds of neuropathy. So that's all I had to talk about now. So hopefully that made sense, and I'll stop sharing my screen and we can
I did a full workup with Dr. Hope about 5 years ago.
Dr. Hope.
How often do you suggest that we go back for follow-up if there's no significant change in anything, symptoms or?
Yeah, I usually leave it up to people based on their comfort, and there's a wide range. I think I usually suggest every year or two, but some people I think really like the every year visit, and some people I think fade away and don't come back unless even after a couple of years, unless there's a change.
Mm-hmm. Right.
Honestly, it's hard right now for us to see have follow-up visits, because there's this crush of people coming back after COVID. So we're sort of getting like 2.5 years worth of new patients trying to come to the clinic. So it's hard.
That's what I actually said to my husband. I said, I don't feel like I should take a place. I am doing fine. So I'm not making an appointment or trying to get an appointment. Yet I have a friend who would desperately like to see you or Dr. Hoke.
Yeah. So that there are, I forget now, for Dr. Hoke has a peripheral nerve research registry that he runs. I forget if they are doing yearly visits for that, for the
I'm in it, but nobody's contacted me, so I presume not.
Yeah. So there's the inherited neuropathy consortium that I'm associated with. When we enroll patients, we do longitudinal studies. So the goal is to see patients every year.
I see.
So in that case, we encourage, just for the sake of the study, to come back. But recognizing that if people are doing well and there's not much change, then we really don't have a lot to offer, other than to it's really you offering your medical history and progression,
Right.
to the medical community to learn more about these conditions.
Uh-huh. But is that taking up someone else's valuable time to do that? I mean, to come back to do that?
Most of us have time slots set aside for new patients and follow-ups. And so, you might, if you try to make a follow-up appointment, you might be told that you can't get one for 6 months or 9 months or something like that.
Uh-huh.
But that doesn't really cut into new patient visits.
Okay.
Okay, Rebecca. Rebecca asks, are there any specific food or drinks that can activate neuropathy, and anything we should specifically avoid?
No, I wish there was a vitamin. I wish there was a food or a diet. Really, all we know is that exercise is beneficial. Regular exercise, kind of a combination of cardiovascular exercise, even some weight training. There's even some data that's coming out kind of comparing brief high intensity exercise versus, less intense exercise. And so these are things that are being actively investigated to try to figure out what exercise is best. But there does seem to be, apart from just, general health of exercise, there's something that helps nerves in particular. And so that's sort of the best treatment we have right now. The other thing is that I think probably many forms of idiopathic neuropathy might actually be due to a combination of maybe not diabetes, maybe pre-diabetes, and then high cholesterol and sort of other sort of metabolic issues, sometimes obesity, or even not just sort of having more abdominal obesity. These things have shown to be, especially when they're combined, risk factors for neuropathy. And so in that case, I think diet and exercise to address the sort of blood sugar issues, and if there's cholesterol issues, those can indirectly help the nerves because they might actually be partly driving the neuropathy.
Okay, the question I have is, I know like the Foundation for Peripheral Neuropathy has a list of medications on there that can possibly cause neuropathy. And this is just a general question, not in regard to any one particular medication, but if a person has neuropathy and they would take a medication that can cause neuropathy, could that possibly make your neuropathy worse or speed up the progression of the neuropathy?
Yeah, it could,
Okay.
in theory. I think you're exactly right. So the same medicines that can cause neuropathy could make it worse. I think that's that list that I think the CMTA puts out, and there may be others, is generally pretty good. I looked into I tried to dig into that literature a little bit more about some of those medications, and some of them, there's really good evidence, some of them not so much. So I think, if you can avoid the medications on that list, that's great. If there's medications on that list that your doctor feels you really need, then it might be worth a conversation with a neurologist to really dig in to see just how good is the evidence for that particular medicine. Because some of them it's a bit iffy, and so it might be a balance of how important is the medicine versus how good is the evidence that it'll make neuropathy worse.
Is there a way to identify a neuromuscular neurologist without looking through their training? Is there a special certification or anything, a list, an association?
So at least for academic places, so Hopkins, University of Maryland, every neurologist is very specialized, and so there would be sort of a neuromuscular division within a given hospital, and you could inquire specifically in that division whether who to see.
Yeah, hi, thank you so much. I have small fiber neuropathy, and I'm wondering about the axonal and demyelinating. I thought small fiber was myelinated and unmyelinated nerves that are affected. And in that case, how does demyelinating and axonal relate?
Yeah, so you're right. Most of the small nerves are not myelinated. There are some that are, but the small fiber neuropathies that we understand, which is not most of them, but there are actually genetic forms of small fiber neuropathy. And so that's mostly an axonal problem, but small fiber neuropathy sometimes is sort of a precursor to other forms of neuropathy, and sometimes it's its own entity where it doesn't sort of spill over to affect other things even after years. And I would say if idiopathic neuropathy is not very well understood, small fiber neuropathy is, you know, even more so. There are some things that we know can cause it, but a lot of times it sort of comes on all at once. Sometimes it progresses, sometimes it sort of stabilizes, sometimes it gets slowly better, but we just don't really understand what's driving it. It's for the most part, it's sort of in its own category, and it's a poorly understood category.
I was pre-diabetic for about at least a decade before I was diagnosed 24 years ago. In the subsequent 10 years, I lost about 100 pounds. I'm no longer pre-diabetic, and yet PN just keeps right on rolling along. And I'm wondering, first off, I guess there's really two questions. First off, do we understand the pattern of neuropathy, of peripheral neuropathy? And I'm thinking about not the hereditary, but more the more common forms. Do we know the time pattern? That is, my impression is that it runs under the unnoticed, typically for a long period of time. You got a tingle in the toes, perhaps, but no major symptoms of weakness, and then it gets much worse, and at some point the damage to the axons is so massive, and the nerves that drive those axons is so massive that you start losing strength very quickly. So that first question is that understood to be the common pattern for regular peripheral neuropathy?
Yeah, all right. So the first question is sort of the normal progression. I mean, we have a general sense, but there's a lot of variability. So even you know, so idiopathic neuropathy is a bit messy because we don't know the cause, and everyone's cause might be a little bit different. But if you take the example of CMT, you know, say we know exactly the same genetic change, and we follow a family that they all have the same genetic change, there's a huge variation in how severe their neuropathy is and how it progresses over time. And so even the sort of the cleanest experiment, there's a lot of variability. So everyone seems to progress at a different rate. I mean, in general, when neuropathy progresses, you get worsening sensation, and hopefully, you know, sometimes that goes with pain, sometimes it's just numbness, but usually only the most severe neuropathies progress to the point where you can't walk eventually. There might be weakness at the ankles, but even patients with CMT that have, you know, kind of bad CMT usually remain able to walk. They might need AFO braces to help stabilize their ankles, but so for the most part, neuropathy doesn't usually progress to the point that people are really disabled. I mean, there are certainly exceptions to that, but there's just a tremendous variability in progression. It's something that a lot of people are trying to address, trying to understand, you know, how certain people progress, and then if there are people that progress more rapidly, why that is and what factors determine that.
I was interested if you've done longitudinal studies to investigate the differences in the progression of persons with CMT and the results compared with persons with acquired PN. Or if you work with CMT, have you done longitudinal studies of family members with CMT?
Yeah, so both are being done. So I've been involved in the studies on CMT longitudinally, and I know others are doing longitudinal studies of patients with idiopathic or other forms. So they're not part of the same study, but they're sort of happening, you know, separately but in parallel. And so that's a big part of the CMT research that I'm a part of in the clinic. And so there's I think now almost 10,000 patients enrolled in the world in these kinds of longitudinal studies to see what happens over time. one of the take-home messages is that it's very highly variable patient to patient, even within the same family. But the real goal of these is twofold: to try to understand what happens over time, but then also to understand how we can measure change over time, so that if we get to the point where we can do a clinical trial, we know what to measure to see if the drugs are effective or not. That turns out to be a really hard thing for neuropathy, because on the, for all of you, it's a benefit that it moves slowly, but from a research perspective, it creates challenges because it's hard to know if the drug is working because it takes so long to see.
And the second question is, since the presumed cause, because I went to Hopkins and they did all their tests, and I've had tests for the last 23 years, and it's all idiopathic at all stages. Is it possible that at some point in life, some part of my body got damaged that's critical for the health of my nerves, and once damaged, there's no way to stop the process?
So, so I think probably not something that would progress. What we know from chemotherapy neuropathy and other sort of toxic neuropathies is that when you're exposed to the toxin, the neuropathy gets bad, and then sometimes it gets worse for a little while. And generally it should stabilize after that. If it keeps getting worse, that probably indicates that there was something else going on in the background beyond that. So the lesson from toxic neuropathies is that you can't really cause ongoing nerve damage from something that happens one time. But if say you had a predisposition to developing idiopathic neuropathy and you get chemotherapy, you might get a neuropathy, and then over time it might get worse because of your own background, your risk factor for neuropathy, but it doesn't mean that the chemotherapy is still responsible for the worsening.
Sure. I was just curious about it and wondered if well, I guess one of my questions would be, is there a defining characteristic of patients with mitochondrial neuropathy? And where can you learn more about it?
No, there's not. So mitochondrial diseases kind of run the gamut of pure neuropathy. So CMT2A is sort of a mitochondrial form of CMT, and they just have, those patients have pretty typical neuropathy that looks a lot like other forms. And then there are other mitochondrial conditions that can cause things like really severe problems with balance, problems moving the eyes, problems with the GI system. And the thing that's weird about mitochondria is they were actually basically bacteria sort of that got into our bodies. So they have their own genome and they have their own way of functioning. And so certain parts of your body can have different mitochondria than other parts. So it can get really complicated. Usually if we think about mitochondrial disease outside of the CMT2A that I've talked about, the clue is that there's multiple different problems going on at the same time. So maybe a problem with muscle, maybe a problem with moving the eyes, maybe a problem with the GI system and neuropathy. And so when you start to get lots of different unexplained systems involved, then that's when people think about mitochondrial disease. And it can be challenging to test for it, because mitochondria are kind of, like I said, these almost like these invaders that are not quite part of our normal genome.
I was just wondering if demyelinating and axonal neuropathy were common to have both forms of neuropathy.
It's in general, no, but the exception to that is in diabetes or what we call other kind of metabolic neuropathy. So kidney problems can also cause neuropathy, and there's something about diabetes and the issues that go along with kidney problems that gives sort of a mixture of demyelination and axonal degeneration. Usually the demyelinating part is kind of mild, but usually if we see a mixed neuropathy of those two types, those are the things that we think about.
Thank you.
But often with demyelinating neuropathy over time, as the myelin becomes unhealthy, the axon can then also get affected. And so it's sort of, the primary problem is demyelination, but then it spills into causing axonal damage as well.
Okay, thank you.
Leads to Ron Medford's question. Can you explain what I can do to get gene testing? I think a lot of us are interested and we're not sure if there's a long wait. We've heard at Hopkins. What do you suggest?
Yeah, I think seeing a neuromuscular neurologist, which is what I am, is probably the best way to do it. So, you know, within neurology, there's lots of different specialties. So people that specialize in neuropathy generally have some kind of neuromuscular training. And I think anyone who is in that sort of subspecialty should be able to make an assessment of whether that kind of genetic testing is indicated and then how to do it.
And the last question related, does insurance typically cover gene testing?
So the nice thing now is that insurance doesn't get involved at all. To explain why, so basically there's a company called Alanylam, which has developed a treatment for familial amyloid polyneuropathy, which is a genetic neuropathy. And they are trying to identify patients that have this genetic form of neuropathy. And so they're funding all this genetic testing for free right now. And so it's a win-win. Means we can get free testing and they can identify patients and make their money. And, you know, we can also identify those patients and treat them. So that's why it's free. So they have this sort of code, the partnership code that you can use when you order the genetic testing. And so that's what all of us
So
do.
the neuromuscular neurologist would just know that, and we
Yeah.
wouldn't have to worry about how to, okay.
If they told you it was going to cost something, you should say, well, I heard that, you know, Invitae is the company that we use. This should be free through Invitae. And it's not like a Hopkins specific program. It's for anyone that would be ordering these tests.
Okay, perhaps you can add that to the chat.
You asked, does good balance and still being athletic rule out hereditary neuropathology or neuropathy as what you have?
Yeah, there's no hard and fast rules of any of this. There are, it makes it less likely, but it certainly doesn't rule it out. So, you know, ways that could happen is you could have a late onset form of CMT where, you know, that's the exception, but they do happen. And then you can have a motor only type of CMT. Some of those genes I mentioned tend to affect strength more than sensation or balance. So it's still possible, but you know, and the nice thing these days is it's easy to get genetic testing. So if there's any question, then, you know, it's easy to get that testing done.
Okay, Rich's question is, there are about 100 genes related to neuropathy that have been identified. How many new genes get added to the list every year?
Yeah, I would say, you know, a few maybe each year, but what's been really remarkable is you would think that as you added more genes each year, you know, you would get, so I showed that list where, you know, CMT1A is almost half of all CMT. And then, you know, some of the other ones are like 10%. And you would expect that maybe as you keep adding ones, you would get, you know, for a while you'd get ones that are like 5% and 2% and 1%. And then you would start be identifying, you know, there's like 2 people in the whole world that have this kind. It's actually not the case. So that sword neuropathy, like I said, was just discovered, and that's actually a big chunk of the pie. And there's been a couple of other genes that were recently discovered that are surprisingly large group of patients at a high percentage. And some of it has to do with just the genes that we're finding now are a little bit, they're harder to find for some reason, that, you know, details I don't need to get into, but they weren't low hanging fruit, even though they account for a larger percentage of neuropathy than you might expect. So it's been surprising. I wouldn't have predicted a few years ago that we would find the most common recessive form of neuropathy. You know, I would have thought that it had already been discovered.
I went to Hopkins in 2014 with EMGs both times and a genetic counselor, and they said that he did think it was CMT, and she said that it the symptoms indicated it was probably hereditary, but I couldn't prove it because I never had any of my ancestors tested, including my parents. I have no siblings. Is it worthwhile to consider retesting after now 6 years, given the fact that I've been to NIH for 4 days in their hospital, which is also free if they accept you, and they came up with a different diagnosis, not CMT, which upset me a bit. So I'm a bit of a quandary there. And that would be, Dr. Chaudhry said afterwards, I had three choices. I could continue to do nothing and learn to live with it, even though it's degenerative, or I could go on a weekly steroid injection, or I could go on I forget the name, but they take the marrow out of your bones, cleanse it, and put it back again. Something lymphophleresis, I think. What can you just comment on if I have CMT2, what is the best thing for me to do now?
Yeah, so
is it worth making the effort to travel in my wheelchair to Hopkins for another test there?
and to take a step back, I think, you know, it's not uncommon that we do full evaluation and we still can't determine whether something's inherited or not. And the reason is, you know, we know 100 something genes, but there's probably many that we haven't discovered. And so, you know, we can't fully rule out CMT by having testing that doesn't show anything, because you could have a mutation in a gene that hasn't been linked to CMT yet. So I think probably what Dr. Chaudhry was getting at is, you know, maybe the main forms of neuropathy that are actually treatable and can kind of get reversed are these sort of inflammatory neuropathies like CIDP. And sometimes steroids help, sometimes IVIG helps, sometimes the other thing you described, plasma exchange helps. So sometimes, usually you can make a diagnosis and you kind of know what you're dealing with. Sometimes it's not so easy. In those cases, you can try one of these treatments and follow things very closely to see if they get better. If they get better, then you say, okay, we'll keep going. If it doesn't, then you say, you know, we're done with this treatment. It didn't work. It's not worth the risk and the cost, et cetera. In your case, you know, when you try these treatments and they don't work, then you do the things that you suggested. So there's unfortunately, no treatment to address the underlying issue, but you sort of deal with the symptoms and try to live with it as best as possible, which is why people like me are trying to study this, you know, more to be able to someday offer more than. I think it's worth reaching out at least. So, you know, we can send these, it's worth having the conversation. So, you know, I don't know the whole details, but if I had a patient who had been tested 5 years ago and I really thought they had an inherited neuropathy and the testing was negative and they sent me an email like you, I would say, oh, let's do it again. And we can actually have the kits, it's a spit kit, we can have it mailed to your home. So it wouldn't even require a physical visit, perhaps. It depends on how your physician likes to run things, but that's an option.
My mother had peripheral neuropathy. My sister has diabetic. Mine is idiopathic. And they did a genetic marker, whatever, and found nothing. Is there any reason, because of new research, to repeat that since it's been 5 years?
Yeah, that's probably worth doing. There's probably 5 years ago, maybe it was like 60 genes or so, I would guess, that would have been on a panel. And now it's a little over 100.
Okay.
So yeah, I mean, the other thing is it's free the way that with the testing that we do. So there's not much downside to it.
Okay.
And it's also helpful, you know, what we often get in these tests is some variants, you know, what they call variants of unknown significance. You don't know whether they're causing disease or not. But if you have other family members that either have neuropathy or don't, then you can also test them, and that can give you some insight into whether a variant is likely to be doing anything, or it's just kind of part of normal genetic variability.
I have a hereditary neuropathy, and my mother had it. I have a sister with it, and okay, so I have 2 children and 4 grandchildren. Is there hope that there will be some sort of way to block the development of a full-blown peripheral neuropathy, you know, expressing itself in the future? Or is this something that I should just say, watch out, it's going to come?
Yeah, I think it depends on the type. In theory, at some point, you know, this genetic editing strategy could really be applicable for every type of CMT and other types of genetic diseases, but that's not going to be on the horizon soon. But I think it's reasonable for some of the types of CMT that are better studied, that there might be something that can really slow it down or stop it. I think for CMT1A and CMT2A, maybe these CMT2C with the TRPV4 mutations, that might be something that, you know, children or grandchildren might have access to. But it really, you know, the other, you know, 100+ genes, maybe not soon. You know, it's just kind of hit or miss on which ones might be treatable with these different, very specific strategies. That's the problem. It has to be very specific for each of these conditions.
Okay.
Although, you know, I mentioned that, you know, that's why something like HDAC6 inhibition is interesting, because maybe it doesn't have to be so specific. But that, you know, it also have a long way to go before that's brought to a clinical trial, I think.
Okay.
You know, if you are fortunate in that you have had a gene that's been identified that is related to neuropathy, you know, what can be done? Is there hope? I mean, once you know that, then what, you know, going forward, yeah, what is there?
Yeah, that's a good question. So yeah, we're talking about really trying to get a diagnosis, but then what's the question of once you get a diagnosis, what do you do with that? It kind of depends on the gene. So some, you know, the ones that are more common have been more studied. So there may be a clinical trial in the horizon in the next 5 years for CMT1A. You know, it's half of CMT, and there's been a lot of work to try to address it. And then, you know, the gene that I mostly work on is this TRPV4 gene, and that's, you know, maybe 1 or 2% of all CMT. But because of the nature of the gene and the fact that there's a drug that can already target it, you know, that might be one that receives more interest and might lead to a treatment more quickly. And then, you know, there may be many others where there isn't any good rationale, at least currently, for how to treat them. And so in those cases, you know, there's not even necessarily someone in the world who's studying that gene with the goal of treatment. So it's kind of hit or miss. It's a bit random, or it sort of might seem random, but to some extent it's based on if a gene is discovered as a cause of CMT, then you look at it and you say, is there some way that we think we could actually treat this? And if there is, then someone might study it. And if there's not, then maybe not.
I guess the less rare, the better, perhaps.
In general, yeah. The other thing I'll say, though, I mentioned the SORD neuropathy, and there's another gene that was recently discovered in the last few years that accounts for a lot of sensory-only neuropathy. And both of those, because they're so new, they're not even yet on those genetic testing panels. And so it's still, there's unfortunately a delay after a gene is discovered before it's easy to test for it.
Thank you. Thank you.
Dr. McCray, so I have CIDP, and I've been told that sometimes that gets confused with CMT, and I'm wondering, is that true?
Okay. So the first question, it should be, there's some times when it's hard to distinguish CIDP from CMT type 1. This goes back a little bit to what I was saying with in the talk about sometimes there's very characteristic changes for CMT type 1. Basically, when you have CMT type 1, all the nerves are slow and they sort of all look the same, whereas CIDP is sort of patchy. And if you compare one arm to the other arm or one leg to the other leg, it looks very different. Even though there's demyelination, it's to different extents. And there's other ways to sort it out, but usually it can be done based on exam, based on history, and based on an EMG.
I'm just going to ask, will you be able to change the gene makeup like they can do for sickle cell anemia in the future?
Probably someday, yeah. So that's a really exciting potential treatment. So sort of gene editing. And I think CMT is a good candidate for that because a lot of these conditions are caused by just a single genetic change. The challenge for CMT is that you need to get the treatment to the right cells. So if it's CMT type 1, you have to get it to the Schwann cells that make the myelin. And if it's CMT type 2, you have to get it to all of the neurons themselves that live either in the spinal cord and send their processes out or just outside the spinal cord. So, you know, it's a little easier to target blood cells like in sickle cell disease, but that at least shows us that this kind of thing is possible. And then the next step is trying to figure out how to get the treatment exactly where it needs to go. But yeah, I think someday that will be a reality.
Dr. McCray, so I have CIDP, and then also I have both motor and sensory. So when I first was diagnosed, it was almost all demyelinating. So, but I more recently got a nerve conduction test, and there was a mix between demyelinating and axonal. And the doctor who was doing the test, and my neurologist, they were all excited about this, but I was not. I'm like, does that mean it got worse? And they're like, no, that means that your nerves are trying to regenerate.
So then I guess, you know, the other part of the question, to have axonal degeneration after, or to have mixed neuropathy. So one thing that can happen if you have CIDP and it's treated, the demyelination can get better, can go away completely. And then, you know, with time, with the longstanding demyelination, then you can get axonal loss as well. So, you know, I don't know in your case, which of those is at play, but, you know, that what you described can happen, and depending on the whole context, it can be a good sign or not a good sign. I don't know specifically in your case. Hopefully that helps a little bit.
Okay.
Yeah, that does help. I'm doing the subcutaneous treatment, and that actually really has improved my strength tremendously. So that's kind of why they're thinking that it's regeneration. But thank you.
Subcutaneous.
Jessica, you've had your hand up a while. Did you
And
have
my other question has to do with Ehlers-Danlos syndrome, but which my son has. And I've read that there's an overlap between peripheral neuropathy and Ehlers-Danlos. My son goes to the clinic at Johns Hopkins. Actually, he's turning 30, which is he's going to time out. He's a type 2, which is just hypermobility. So basically, for anybody here who doesn't know, it's basically it just has very hypermobile joints. You know, I've read that then that nerves are able to go through the joints more easily and then being trapped. Is there anything that you can bring to light about this topic?
Yeah, I know there's definitely an association between the two. And I think that's pretty well described, but the underlying reason for that is not, I mean, usually it's a small fiber neuropathy with Ehlers-Danlos. And so it's probably not so simple that it's just sort of the, nerves passing through certain narrowings like in the wrist, because it seems like the neuropathy is more widespread. So there's probably something about whatever causes the joint hypermobility that also may affect the actual structure of the nerves and put them at risk for, being unhealthy over time. But I think the genetics of Ehlers-Danlos are very complicated and not well understood yet. And to my knowledge, there's no gene that directly causes both. It's just an association that, we know that people who have small fiber neuropathy have Ehlers-Danlos and vice versa.
Is there anything to do then? I mean, exercise, just wait for symptoms to develop is sort of an odd way to go about it, but that could be it.
Yeah, there's no specific therapy. I think with small fiber neuropathy, the biggest thing is usually dealing with discomfort or pain, because generally it doesn't cause so much problems with balance and so much problems with strength, but the discomfort and pain is the major thing. And that at least we don't have great medicines, but we have a lot of different medicines that can be tried to help with pain.
Thank you.
Well, thank you very much. I think we got to everyone's questions at this point, and we really appreciated your coming. And perhaps someday you can come back again and give us an update on what's happening with your research. Thank you.